We were discussing about the amino acids in the last class, about the hydrophobic amino acids, about the polar amino acids, and then there was charged amino acid, where positively charged and negatively charged amino acids were there, right? So now first let us understand the hydrophobic amino acid. So the hydrophobic amino acids, also known as aliphatic amino acid, right? So the very first that we have is glycine. As we know, the R group A here is hydrogen, and it is found in flexible parts of protein because it is less bulky. Flexible parts of protein. If you remember the structure, that is NH3 group is there is alpha carbon, there is carboxylic group, and the R group here is the hydrogen, and there is another hydrogen, right? So due to this R group hydrogen, so this is less bulky. This is less bulky. And it is not chiral. Just remember that. I am repeating it again. It is being asked in IIT JAM exam as well as in GATE exam. So you must remember this. So I'm highlighting this part. The glycine is not chiral. Okay. And it can be modified. Just remember this. And it can be modified by addition of fatty acids. So for example, there is myristate of 14 carbon fatty acid. You can change the overall this amino acid just by addition of this particular fatty acid. Okay. Now coming for alanine. The second is alanine. It is called the model amino acid. Model amino acid. The R group here is CH3. So the NH3 positive, C alpha, CO negative, H, and the R group here is CH3. Third is valine. And all of these are used in like hydrophobic part. Hydrophobic part of protein. So the hydrophobic part, so since our cell contains water, right? So cell contain water. So when a protein fold, so there is a hydrophobic core that is formed. Hydrophobic core, and then there is a hydrophilic outer core. Hydrophilic outer core or portion, that you must remember. So the inner part will always be the hydrophobic, will always be formed by the hydrophobic amino acids, right? Third is valine, leucine, and isoleucine. You know why these three are learned together? Because these are also called one of the essential amino acid called BCAA. So these are called branched chain amino acids. And why these are called branched chain amino acids? So you need to go to the previous lecture where I talked about this. So this is leucine. As you can see, there is CH2, CH, and then there's a branch formation, right? So for isoleucine, there's CH, the CH3, and there is CH2 and CH3, right? And for the valine, there is there is CH and CH3. So there is some sort of branching that is happening. So that is why these are called branch chain amino acids. Okay, and you must be knowing about the supplement industry. If not, so these are labeled as muscle protein synthesis booster, because leucine is the most abundant amino acids find in muscles. Again, remember this is the important part. Okay, and the valine, leucine, and isoleucine increase the muscle protein synthesis, that we are going to talk later in this section. Just remember that. Okay, another one is methionine, although I have discussed in the last class. So it contains sulfur. So it can interact with metal ions, interact and bind with metal ions, but it is not included in the bond formation. Just remember that it can interact and bind with metal ions, but it cannot make any sort of bonds by the sulfur. Okay, and it is often found in metalloproteins. Metalloproteins. Another is proline. It is called amino acid because of the group. It affects the protein folding. It is usually found at bends in protein 3D structure. So we are going to learn about the primary, secondary, tertiary, and quaternary structures. At that point of time, I'm going to explain it well. But just remember, so when a protein is folding, right? So if a rope is there, right? So if we have to fold the rope, right? So during the folding, so there should be a flexible part, right? Or there should be a rigid part that is this shape only, right? So at that point of time, we are going to keep that particular amino acid at that thing, right? So same thing happens during the protein folding. So whenever there is a bend in the protein structure, right? So at those places usually proline is found. Just remember the rope thing. Okay, so we have to bend the rope. So either this part should be flexible. So for the flexibility, either glycine will be there or it should be rigid, so proline would be there. Just remember that during bends or during a protein folding, so glycine and proline usually help during the bending. Okay. Also, hydroxylation, remember this part, of proline is important for structure of collagen. Structure of collagen. I'm going to highlight this part because this might be asked, because question from the collagen usually come, and proline plays a very important role in it. Okay. Now we are going to learn about the aromatic amino acids, also called aromatic hydrocarbons, right? So like I said, so there are phenylalanine, there is tryptophan, and there is tyrosine. All three of them help in protein quantification. So for example, if I have a solution of 1 ml and I want to know what amount of protein is present in this particular solution, now based on these three amino acid I can find that. Okay, and why is that? Because these three are fluorescent in nature, and how they are I'm going to explain in a bit. So they can absorb UV. Okay, so they UV absorb at 250 to 300 nanometer, or technically at 280 nanometer, due to which what happens is we can actually identify the particular amount of protein in any solution. Okay, and tryptophan is one of those amino acid which is converted to serotonin. And what is serotonin? If you guys know, it gives us a pleasant feeling. So love हार्मोन जिसे बोलते हैं ना, कि वो लव वाली फीलिंग जो आती है, that is because of serotonin. So it has a sedative effect, and this question was asked during 2020 jam preparation, jam exam, not preparation, 2020 jam exam. So there they asked question about the serotonin pathway, melatonin pathway, as well as this tryptophan pathway. So they directly ask like which is made by the serotonin is made by which particular pathway. So you must know about this. Okay, so the extreme high level of this serotonin can cause a maniac state. So if your girlfriend or your boyfriend is maniac, so just remember they have high level of serotonin. And when I discuss about the part where they can actually quantify the protein, and it is because of the fundamental of UV spectroscopy. So if you guys do not know about the UV spectroscopy, so I'm just making a diagram for you. So there is this benzyl group and then there is OH. Okay, so this is tyrosine, right? So for example, we have this particular amino acid and this particular amino acids are in the solution, right? So I have a particular source of light here. So let's suppose this is light, right? And a particular wavelength of light is going to hit. So let's suppose that is 280 nanometer of wavelength. So let suppose there is 100% of incident light is coming, so I'm going to label it as IL, and there is some sort of absorption that is going to take place, right? So this absorption or the absorbance is happening, and then there is a transmitted light, so it is called IT, right? And then there will be a detector which is going to detect the light, and based on that I'll have a output, that how much incident light has been absorbed to the solution and how much it has been transmitted, right? So there's a formula called A equal minus log of IT divided by IL. Okay, and this directly correspond to A concentration as well as the path length. So this is called B-Lambert's law that we are going to discuss in very detail in upcoming slides. But what is actually happening is, so incident light is getting here, right? So the electrons are in an excited state, and due to which there is a higher electronic state, that due to which there is a formation of resonance here. Okay, and due to this resonance, what happens is, so when a electron jumps from lower state to a higher state, okay, and when it comes from higher state to a lower state, so it releases some sort of energy, right? So that energy is called fluorescence or any sort of energy. So like there are multiple different energy, but in this case this is a fluorescence energy, and we detect this particular energy during this detection. Okay, and based on this we can actually quantify what is the actual amount of amino acid is there. Now the question may arise, sir, so what if a protein only contains tryptophan, phenylalanine and this tyrosine, right? Tyrosine. And what if, so in one case these are only present there, and in another case these are totally absent, right? So